students, I am Dr. Divya on behalf of TeenEinstein. So now we are in the second chapter, sexual reproduction in flowering plants. Correct? So in sexual reproduction of flowering plants, so we all know that plants will reproduce sexually, and flowers are the sites of that sexual reproduction. So generally plants we will use for decoration. In all major occasions we'll use flowers, right, to attract people. See, flower means that is a kind of attraction. So almost all the flowering plants shows sexual reproduction. Then there are different types of inflorescence also. There is a great diversity in the structure of the inflorescence and flower and the floral parts that will show a wide range of adaptation, right, and that will form some kind of end products. Like after sexual reproduction, the end product of that is seeds and fruits. So in this chapter, sexual reproduction in flowering plants, we are going to discuss about the morphology, structure and the process of sexual reproduction in flowering plants, especially flowering plants or angiosperms, right. So in this, under the sexual reproduction in flowering plants, flower, what is a flower and the floral parts, we are going to discuss about that. We have already discussed those things in the previous classes. Then fertilization, that is a major event, fertilization. So before fertilization, how a flower it is getting adapted for a fertilization, or how they are making arrangements for the fertilization. Then the fertilization process, that also we will learn. Then after fertilization, what is going to happen, or post fertilization events. So those and all included in this topic. So I'll give a brief introduction about the chapter and just main points of previous classes. Okay, a quick review I'll give. So all flowering plants will reproduce through sexual reproduction by various features. Which and all those features? They have colorful flowers, then pollen grains, they have nectar, all these and all various features of a flower. This pollen grains, it is carried by insects also. So they are the agents of pollination, insects, right. Why insects are coming to a flower? For the nectar. And for attracting these insects, flowers will become colorful. There are flowers which are not colorful and small flowers also there, but most of the cases this is the reason. Then flowers are formed in the shoot apices or at the tip through structural and physiological changes induced by hormones, and that serves as the site of reproduction, right. And we discussed that a complete flower, it is having calyx, corolla, stamen and carpel, like that 4 walls. We discussed about calyx, corolla, stamen and carpel, right. Calyx means sepals. And what is the role of this sepal? To protect inner whorls in bud condition. When that flower is in bud condition, or it is in bud condition, means that time this calyx will protect the flower. Here you can see, if it is in bud stage, it will protect the flower. And next, corolla. Corolla means petals, a collection of petals. This helps in the pollination and protection of this inner whorl. Inner whorls are protected with the help of calyx and corolla. Corolla means petal, to help in the pollination. How corolla is helping in the pollination? Corolla, in most of the cases, corolla will be colorful. So that will attract the insects or pollinating agents. Okay. So outermost whorl is calyx, and next to that, inner to calyx is corolla, right. And this calyx and corolla, they are called as non-essential whorls. Why? These are called as non-essential whorls. Why it is called as non-essential whorls? Because without the help of this calyx and corolla, also reproduction will take place. Reproduction needs male and female part, right? So next we are going to speak about essential whorls. So these and all we have already discussed. Essential whorl means andrecium and gynoecium. This andrecium and gynoecium are called as essential whorls. Okay. So the essential whorl, andrecium means stamen. And the stamen consists of anther, then connective and filament. Stamen, it is having three parts: anther, connective and filament. And this helps in the reproduction as a male reproductive organ. Okay. So andrecium helps in the reproduction as a male reproductive organ. Then gynoecium. Gynoecium is carpel. Okay. It is a collection of carpels. It is called as gynoecium. And this carpel consists of ovary, style and stigma. In this you can see ovary, style and stigma. That is called as carpel. So carpel helps in the reproduction as a female reproductive organ. What is the role of carpel? It is a female reproductive organ. So essential whorls, andrecium and gynoecium are called as essential whorls because that is a male and female organ. Then non-essential whorls are calyx and corolla. Okay, sepals and petals. Okay. So that is the structure of a complete flower that is having four whorls: calyx, corolla, andrecium and gynoecium. In this, calyx and corolla are non-essential and andrecium and gynoecium are the essential whorls because they are the male and female reproductive organ. Okay. Then the whole process of this sexual reproduction in flowering plants can be divided into three steps. Pre-fertilization, that in this chapter we are learning about the pre-fertilization structures and the events. Then double fertilization. In the fertilization step, double fertilization is taking place. Next is post-fertilization. Post-fertilization, under post-fertilization title also we will learn about structure and events. Okay, so the whole process of sexual reproduction in flowering plants can be divided into three steps: pre-fertilization, then fertilization. Fertilization in the sense that is a double fertilization. Then there two fertilizations are taking place. That's why it is called as double fertilization. And next is post-fertilization. After fertilization, the structures and events coming under this. Those things we are going to discuss. Then pre-fertilization structures and events. So we have discussed about these pre-fertilization structures and events, and these events which occur before fertilization or the preparation for fertilization, that is called as pre-fertilization events. So it is further categorized as pollen grain formation, then embryo sac formation, then pollination and pollen pistil interaction. Okay. So pre-fertilization structures and events, under that pollen grain formation is there, embryo sac formation, then pollination, then pollen pistil interaction. So all these things are coming under pre-fertilization structure and events. So first we learned about pollen grain formation. So pollen grains are formed in pollen sacs of anther, and this anther is bilobed. And the lobe encloses 4 pollen sacs or microsporangia. Okay. So anther, it is bilobed and each lobe consists of 2 microsporangia. So in total that is having 4 pollen sacs. Okay. Next, structure of microsporangium. Microsporangium or pollen sac that we have learned, that consists of 4 layers, right? So the microsporangium consists of 4 layers: epidermis, endothecium, middle layer and tapetum, right? Epidermis, endothecium, middle layer and tapetum. Epidermis, that is a single layer, and the outermost layer, and that acts as a protective covering. Okay. Next is endothecium. Endothecium is also, that may be single or multi-layer. Okay, that sub-epidermal layer, that is the sub-epidermal layer, and that helps in the dispersal of microspores. Okay, then comes the middle layers. Middle layers may be bi or tri-layered. 2 layers of cells or 3 layers of cells may be present in middle layers, and this is subendothecium layer. Which one? Middle layer. It is a subendothecium layer, and this stores the reserve food during the early stage of the microspore development. Okay, in the early stage of microspore development, this middle layer store reserve food, and the last layer is the tapetum. Tapetum is unilayer, and that is an innermost layer, and that forms nutrition for pollen grains. For the developing pollen grains, they are getting nutrition from tapetum, and that secretes pollen kit and tryphin substances. Okay, these are the important MCQs. Nutrition is getting from tapetum, the layer tapetum, and it secretes pollen kit and tryphin substances. Okay. So microsporangium consists of 4 layers: epidermis, endothecium, middle layer, and tapetum. Next, we discussed about microsporogenesis, or the formation of microspore. Okay, how microspore is formed? So a young anther, when we are considering about a young anther, that has homogeneous mass of hypodermal cells, right, and that is bounded by a single layer, that is covered by a single layer of epidermis. Then below this epidermis, archisporial cells are present. These archisporial cells are having a prominent nucleus and so much of cytoplasm, abundant protoplasm is present. Okay, so below the epidermis, archisporial cells, that is having abundant protoplasm plus prominent nucleus. Then this archisporial cells are present, I said, and that goes to periclinical division, and that forms outer group of parietal cells. An outer group of parietal cell that forms an inner group of sporogenous tissue. So that undergoes periclinical division and forms an outer layer of parietal cells and inner layer of sporogenous tissue. Okay, and the cells of these parietal cells that divide by anticlinal and periclinal division. That is very important, anticlinal and periclinal division, and that forms 2 to 5 layer anther wall. At the same time, the sporogenous tissues, sporogenous cells, that will divide mitotically, and that forms microspore mother cell. Okay, this parietal cells that will undergo anticlinal or periclinal division, and it will form 2 to 5 layer of anther wall. At the same time, the sporogenous tissue, the cells of the sporogenous tissue will undergo meiotic division, and it forms the microspore mother cells. And this microspore or pollen mother cell, microspore mother cell or pollen mother cell, divides by meiosis. Okay, first division it is mitosis, then microspore or pollen mother cell divides by meiosis to form 4 haploid microspores or pollen grains. Okay, this is called as microsporogenesis. Microspore mother cell divide by meiosis to form 4 haploid microspore or pollen grain. It is in the tetrad form. Okay, this process is called as microsporogenesis. Okay, so the microsporangium then that nourishes the developing pollen grain. Then microspore mother cell then forms the microspore tetrad. Then each microspore, this microspore tetrad, it will get separated. Then each from that each cell, it is having a vegetative cell which is bigger and with abundant food material. Then it is having one generative cell which floats in the cytoplasm of the vegetative cell. Okay. Then pollen grain. Pollen grain is having, and each forms the pollen grain, and pollen grain is having two layers covering intine, inner intine and outer exine. This intine is made up of cellulose and pectin material. At the same time, exine is mainly made up of sporopollenin, that most resistant material, organic material. So it is discontinuous at germ pore. Okay, this exine covering is not everywhere. It is there, a gap will be there in exine, that is called as germ pore. Through that, pollen tube germinates. Okay, so that is the structure of a mature pollen grain. Then embryo sac formation. That embryo sac formation occurs in the ovule, and this ovule develops into seed after fertilization. Then that embryo sac formation include two processes, megasporogenesis and megagametogenesis. Okay. Next, we learned about the structure of ovule or Megasporangium. So the structure of ovule or Megasporangium, we have learned all this, but before today's class, I want to remind you about the structure of this ovule, because we are going to learn about different types of ovule. So the ovule, it is a very small structure. Here you can see a small structure, and it is attached to the placenta. Okay, placenta by a stalk, called a stalk-like structure, that is called as funicle or funiculus. This is general structure I am explaining. It is attached to the placenta by funicle or funiculus, and the body of the ovule fuses with the funicle at a particular region. That region is called as hilum. Here you can see the body of the ovule that fuses with the funicle, that is connected to the funicle at a particular region. That region is called as hilum. And this hilum represents the junction between ovule and funicle. So each ovule has one or two protective envelopes called integuments. In the last class we said integuments, one or two layer of protective layer of cells, that is called as integuments. And this integuments encircle the nucellus. Right? This integument, this is the nucellus. This integument encircles the nucellus, and at the tip, or except at the tip, and at the tip there will be an opening. This is called as micropyle. See here, everywhere these integuments are covered except at the tip. This is called as, this gap is called as micropyle. And opposite to the micropyle, see where micropyle is present, that side is called as micropylar region. And just exact opposite to this micropyle or micropylar end, chalaza is present here. This is chalaza, and this represents chalasal end. Okay. So enclosed within the integuments, a mass of cells you can see, that is called as nucellus. So within the integuments you can see nucellus. This is a mass of cells, and this nucellus, it is having abundant food material, and in the nucellus this embryo sac or female gametophyte is located. Then an ovule, it is generally, it is having a single embryo sac formed from the megaspore. Okay, single embryo sac formed from the megaspore. Then structure we learned, and in the last class we have learned about two classifications of ovule also. So that classifications was based on the development of nucellus, and second classification was based on the integuments or the number of layers of that integuments. So first one was based on development of nucellus, and second classification was based on number or layers of that integument. So what is ovule? The plant ovule is a part of female reproductive organ, right? So in this part, or in this part of plant, this female reproductive cells are formed and kept, and after fertilization these cells forms the seeds, and later these seeds turn into plant, or it will form a complete plant. Now next classification of ovule is based on the relationship of micropyle, chalaza and hilum. So today's class we are going to start with another classification of ovule. This is based on the relationship of micropyle, chalaza and hilum. And with the body it is connected, with the body of the ovule. This micropyle, chalaza and hilum, it is connected with the body of the ovule and orientation of the funiculus. There are 6 different types of ovule found in angiosperm. So based on that there are 6 different types of ovule. Okay, so micropyle, chalaza and hilum with the body of ovule. The connection between micropyle, chalaza and hilum with the body of ovule, and based on the orientation of the funicle. Based on the orientation of funicle, there are different types of ovule in angiosperms. So based on this, there are 6 types of ovules. First type is atropous or orthotropous. Atropous or orthotropous. In this, the body of the ovule. First one is atropous or orthotropous, first type. And in this, the body of the ovule is upright in position. You can see this is upright in position. It is straight. And this is not a curved type of ovule. And the micropyle, here you can see the micropyle. Where is micropyle? The opening, this is the micropyle, hilum, and these lie in a straight line. Here you can see in this. Okay, this lie in a straight line. So this type of ovule, it is called as straight or upright ovule. There is another name for this, straight or upright ovule. So you can see a atropous ovule or orthotropous ovule. In gymnosperm you can see, then beetle, piper, polygonum, etc. So in this most primitive type of ovule, among all the 6 types, the primitive type of ovule is atropous. Okay. So the main ovule body, it is in upright position. Here in the picture you can see it is in upright position, and the micropyle, chalaza and hilum, they are placed in a straight line. Okay. So examples are beetle, piper, then polygonum, and in gymnosperms also you can see atropous or orthotropous ovule, and it is the most primitive and most simple type of ovule in angiosperm. So which is the simple type of ovule in angiosperm and most primitive type? Orthotropous or atropous ovule. That is the most primitive type of ovule in angiosperms. Next, which type of ovule is present in angiosperms? Next one is hemitropous or hemianatropous ovule. Okay, hemitropous or hemianatropous ovule. So this classification is mainly based on position and relationship of micropyle, then chalaza and hilum with the body of ovule. Okay, so based on that this classification is done. So in the hemitropous or hemianatropous ovule, the body of the ovule bent on funicle at 90-degree angle. Okay, so in this hemitropous, here you can see in hemitropous ovule, the body of the ovule, here this is the body of the ovule. So that ovule body, it is bent on the funicle. This is the funicle or funiculus. So it is bent on the funicle at a 90-degree angle. 90-degree angle. That means the body of the ovule present at right angle to the funicle or funiculus. Here you can see the body of the ovule is right angle to the funicle or funiculus. So therefore hemitropous ovule is intermediate type between orthotropous and anatropous ovule. So the hemitropous ovule is intermediate type between orthotropous and anatropous ovule. Then this type of ovule, it is also called as horizontal ovules because of the position. It is horizontal in position. So this type of ovule is also called as horizontal ovules. Why it is called as horizontal ovules? Because the body of the ovule is present in horizontal position in the funiculus. Okay, here you can say it is in a horizontal position in the funiculus. So in this type of ovule, you can see in the picture, in this type of ovule, micropyle and chalaza, these are present in same line. Here you can see the micropyle, here this is a micropyle and chalaza, they are in the same line. But here one point you need to note down is this micropyle. Micropyle is away from the ovule. Okay. Micropyle, chalaza are present in same line in this, but micropyle is away from hilum. So what is hilum? What is hilum? The junction between the body of ovule and funicle. That is called as hilum. Here in this examples are ranunculus, primula, then golfimia. Okay. So hemitropous or hemianatropous ovule, in this the main ovule body is bent on funiculus at right angle 90 degree. Then this is intermediate type between ortho and anatropous ovule. And this ovule, it is also called as horizontal ovule because the ovule body, it is present in the horizontal position of funiculus. Next is anatropous ovule. Next type is anatropous ovule. So this is the main type of ovule found in most of the plants. Anatropous ovule is the main type of ovule found in almost all the plants. That means most of the angiosperms, in almost all the angiosperms, that means around 80% of angiosperms have this type of ovule. And in this type of ovule, in this type of ovule, in anatropous ovule, this is it is mainly found in angiosperms, almost all the flowering plants, around 80%. And in this type, the body of the ovule, you can see this structure already we have learned in the structure of ovule. Okay, so the body of the ovule is turned at 180-degree angle. Here you can see it is turned at 180 degree. This is a 180-degree angle, it is turned. So this type of ovule is called as inverted or downward oriented ovule. See, in abstract first structure which we studied, in that everything is in a straight position. The micropyle, chalasa and all in a straight position, right? Here it is 180-degree turn. So this type of ovule is called as inverted or downward oriented ovule. So in this type, you can see in the diagram, the micropyle and the chalasa. Micropyle and chalasa are in a straight line, right? This is chalasa, this is micropyle. So it is in a straight line. But in this type, hilum and micropyle, that is side by side. Here you can see hilum and micropyle. This micropyle and chalasa, they are in straight line, and hilum and micropyle are side by side. So this is the most common type of ovule. So this is considered as a typical ovule structure of angiosperms. This is the most common, that's why it is considered as a common type of ovule structure in angiosperm. So already I told nearly 80% of the angiosperm have this type of ovule. That means anatropous ovule. But in capsula, in this also exceptions are there. In capsula, it is absent. In this ovule, micropyle is facing downward condition. In capsula, it is a type of herbaceous plant and it is a biennial plant. And the name capsula means, it came from Latin word, which capsa means a box or a case or a covering. Okay, the capsula came from Latin word, that in that the word capsula, that means box or a case. So the fruit of this, it looks like a wallet. So the capsula, in capsula, anatropous ovules are not present, and the fruit of this capsula looks like a wallet. Okay, so in members of Solanaceae, Cucurbitaceae, etc., all this type of ovules can be found. The ovules of this type can be found in the members of Cucurbitaceae, Solanaceae and all. Then it is this type of ovule, it is also called as resupinate ovule, means upside down ovule. Resupinate or upside down ovule. So what and all we learned about anatropous ovule? The main ovule body is completely turned at 180-degree angle due to unilateral growth of funiculus. And this ovule, it is also termed as inverted or downward oriented ovule. And the chalaza and micropyle, they are arranged in a straight line here. In this also, chalaza and micropyle are in a straight line. And the hilum and the micropyle, they are located side by side in close proximity to each other. An anatropous ovule is found in several angiosperm families. Around 80% this anatropous ovule is present, but it is absent in Capsella. This is the most common type of ovule and it is considered as a typical ovule of angiosperms. For example, members of family Malvaceae, then Cucurbitaceae, Solanaceae and Compositae. Okay, next is Campylotropous ovule. Next type is Campylotropous ovule. That means the ovule is curved. You can see the ovule is curved. Overall the ovule is having a curved shape. It is curved. Then obviously the micropyle and chalaza, when the ovule is curved, micropyle and chalaza will not come in a straight line, right? The nucellus also be seen in curved position. So along with the ovule curve, the nucellus is also seen in a curved position. But embryo sac is in straight position. Here inside the embryo sac, you can see this is the embryo sac, that is in straight position. Nucellus is curved, along with that this ovule is also curved. Okay, but the embryo sac is in straight position. So in the diagram you can see micropyle, chalaza, or this micropyle and chalaza. Actually, usually this will come opposite to each other, right? In other types and all. Here micropyle and chalaza, these are coming nearby and that is near to hilum. In which type? In Campylotropous ovule. So in this classification, whenever you can explain while explaining the structure of ovule, you have to mention the position of chalaza, then micropyle, funicle, and how all these are related. That is the important thing. For board exam, they will ask many questions like different types of ovule. If they are asking based on the structure of ovule, you have to mention the position of chalaza. Okay, because mainly based on the position of chalaza, micropyle and funicle, we are describing about different types of ovule. So mainly these things should be marked. Okay, that means whether they are near to hilum or far away from hilum. That and all we need to mention while writing about these ovule types. And this type of ovule you can see in Leguminosae family and Cruciferaceae family. Which type? In Leguminosae and Cruciferaceae family, this type of ovule can be found. And in Capsella you can see this type of, we said in Capsella is a exceptional case in the case of anatropous ovule. So in Capsella we can see this Campylotropous ovule. Okay, so we said already in Capsella ovule micropyle, it is facing downward condition. So this type is also called as curved ovule. It is also called as curved ovule. The ovule, in total the ovule is curved, but here the embryo sac is in straight position. Okay, that you should not forget, embryo sac in a straight position. But here micropyle and chalaza both are not coming in straight line. Here micropyle and chalaza, it's coming nearby and it is near to which part? Near to hilum. Okay, so the main ovule body is curved so that the micropyle and chalaza are not arranged in a straight line in this. And the nucellus, it is present in the curved position. Nucellus also you can see there is a curve. But the embryo sac remains straight. I said the embryo sac, there is no curvature for this embryo sac. And micropyle is in close proximity to the hilum. Micropyle is very near to the hilum, and members of the family, Leguminosae family, then Caparidaceae, Cruciferaceae. In all this, we can see Campylotropous ovule. Next is Amphitropous ovule. Amphitropous or transverse ovule. Okay. Amphitropous or transverse. So in this type, effective curvature you can see in the nucellus. Here the nucellus part is having that curvature. Then due to the curved effect, due to that curvature in that nucellus, the embryo sac is also, that will also become curved, or it is in horseshoe shaped. In which type of ovule? In amphitropous or transverse ovule, the embryo sac is also in a curved shape. So in this type, hilum, you can see here hilum and micropyle. Hilum and micropyle comes very near, and this is also called as transverse ovule. This is also called as transverse ovule. For example, poppy, that means papaver, then mirabilis. All these are found in transverse ovule. So the curvature of the main ovule body is more effective in the nucellus area, such that the embryo sac becomes horseshoe shaped. Then micropyle is placed near to hilum. In the structure, from the structure itself, it's clear that micropyle is very near to hilum. For example, this type you can see in lemna, poppy. Then members of the family Butomaceae. Okay. And next type is Circinotropous or coiled ovule. Okay. So in this, coiled ovule is present. And in this type of ovule, body of that ovule becomes inverted. Here is exactly inverted. And again turned into straight position due to the growth of the funicle. Okay, first we learned about the first type of ovule. It is in the straight position, but here this is turned completely and made a straight position on the funicle. So I'll explain more clearly. So the body which is present on the funicle, it is at 360-degree rotation. So it is in a 360-degree angle. So I'll make it more clear. The whole body of the ovule is surrounded by funicle. Okay, here you can see the whole body is surrounded by funicle. So in this type, micropyle or micropylar region is away from the hilum, and you can see in the diagram also, you can see the whole body is covered with the funicle, or such a curvature is present. It's around 360-degree rotation. So in this type, micropylar region is away from the hilum. You can see here micropyle is away from the hilum. And Cactaceae family and Opentia family, you can see this. In Cactaceae and in Opentia, you can see this. Next comes the megasporogenesis. This is the megasporogenesis. So already we have learned different types of ovule, right? So what is megasporogenesis? It is a formation of megaspore. Genesis means formation. So it's a formation of megaspore from a megaspore mother cell is called as megasporogenesis, right? So for the formation of this megaspore, we need a megaspore mother cell first. From a mother cell it will form. So we need a megaspore mother cell, and it is called as, from megaspore mother cell, megaspore is forming, that is called as megasporogenesis. So for the formation of the megaspore, we need a megaspore mother cell, that is called as MMC, short form megaspore mother cell, MMC. So we don't know, or we don't have any idea about megaspore mother cell. So first we need to learn about a megaspore mother cell and it is developing, how it is developing, correct? So now let's see how a megaspore mother cell forms. Okay, so you have already learned that a typical pistil that consists of an ovary, style and stigma, right? A typical pistil consists of ovary, style and stigma, right? So inside the ovary there are one or two ovules or one or more ovules, that is called as megasporangia, right? So inside the ovary there are one or more ovules or megasporangia. So in an ovule, okay, in an ovule the main body is called, or the main part is called as nucellus, right? So in the structure itself it's clear, the main part is nucellus, and this nucellus is made of parenchymatous cells. We have already discussed nucellus is with parenchymatous cells, and it is having parenchyma cells, right? And nucellus is made up of parenchyma cells, and these are homogeneous tissue. All cells are similar, homogeneous tissue. So at the nucellus is covered with integuments. These nucellus, it is covered with integuments. Here you can see this is the first one and this is the second one. This is the outer integument and inside this is the inner integument. So this integuments may be either one or two layered, and the integument, it is not completely covering the nucellus, and that surrounds the nucellus all around except one region, right? That region, that is the micropyle. Here we have already mentioned, this is the micropyle. It is covering almost other places, but except micropyle it is covering everywhere, right? So now I have explained the structure of ovule, right? So do you know about the starting stage of this ovule? The ovule at first, they rise as a primordia. That means, primordium means a tissue in the early stage. That is primordium. This is a term used in embryology mainly. So study of embryo, that is called as embryology. So during in this embryological studies, this primordium, that word is used. The primordium is an organ or a tissue in the earliest stage, earliest recognizable stage of a development. Okay, primordium means the organ or tissue in its earliest recognizable stage of the development. So the cells of this primordium, they are called as primordial cells. The cells of the primordium is called as primordial cells, and the ovule, ovule rises first as a primordium on the placenta in the cavity of ovary, where it forms in the cavity of ovary. Then due to the meristematic activity means again due to the divisions, these primordial cells so the primordial cells will be having meristematic activity. So the protuberance or the outgrowth becomes prominent. It will grow more and it will become prominent and forms a nucellus. Okay. Then after nucellus formation, this integument forms and covers the nucellus. First nucellus will form, and after nucellus formation integuments will form and it covers the nucellus. Now nucellus formation happen, right? So hope up to this everything is clear. And next the megasporogenesis begins with the differentiation of archisporial cells. Where it will differentiate in the nucellar hypodermis. Okay, so not from the epidermal cells of nucellus, it's from the hypodermal cells of the nucellus. It develops from the hypodermal cells of the nucellus, and any one hypodermal cell of the nucellus that will differentiate and that increases the size. Okay, that increases in size and that forms specific nucleus, and the cells becomes different from rest of the parenchymatous cells. Okay, and this specific cell is called archisporial cells. That is called as archisporial cells, and these archisporial cells or archisporial cells or archisporium, this is important MCQ, that is a specific cell, parenchymatous cell, which becomes enlarged. So the archisporial cells or archisporium divides mitotically and it forms a primary parietal cell and primary sporogenous cell. Okay, and the primary sporogenous cell that will directly act as a megaspore mother cell. Primary sporogenous cell directly act as a megaspore mother cell, and this will be present at the micropylar region. This megaspore mother cell will be present at the micropylar region. See, here you can see micropylar end. So this megaspore mother cell will be present at the micropylar region. A single microspore mother cell, a single microspore mother cell or a single megaspore mother cell, it is differentiated in the micropylar region of the nucellus, desiring development of ovule. And this megaspore mother cell, it is a large cell with dense cytoplasm. We know megaspore mother cell is a large cell with a dense cytoplasm. So it has a prominent nucleus. You can see a big nucleus it is having, and this differentiated cells of nucellus then undergoes meiotic division and it forms 4 haploid megaspores. Here it will undergo meiotic division and it will form 4 haploid megaspores. First meiosis, it will undergo meiosis 1 and meiosis 2. Okay. So after meiosis 1 means prophase, metaphase, anaphase and telophase and all you have learned. So after meiosis 1 all will be in dyad stage. Then meiosis 2 will occur. This happens as a result of megaspore tetrad formation. So it is in a 4 cell stage. Okay, here megaspore tetrad in a 4 cell stage. Clear? So as a result of meiosis, megaspore tetrad will be formed, and from megaspore mother cell a megaspore tetrad is formed. So what is megasporogenesis? It's formation of megaspore from the megaspore mother cell. Clear? Is it clear? So that's all. And next, how embryo sac will develop, that we will learn in the next class. Now that's all about this topic. So we have learned about megasporogenesis, right? The formation of megaspore from the megaspore mother cell. So that's all. Hope everything is clear. Thank you.